So as I said, the first example was sort of one of the earliest cell simulations. You can, that had only like 25 reactions that you had to look at, but gave you the flavor of stochastic gene expression and how you put in the data. Now let's sort of increase the complexity. We're going to try to mimic the, come up with a model for ribosome biogenesis, one of the biggest blocks of the genetic information processing. And as I said, we are indebted to the work that Carl Rose had done. We know all about the mechanism, and we have looked at this problem several different ways, trying just to look at the assembly of the small subunit. There's an article in Nature where we worked with 2 experimentalists, and then most recently in Nature Communications, 2017. But it took so long just doing the MD simulation, we thought, well, we'll never be able to do the 3,000 that Wolfgang Baumeister is measuring in his systems, even for slow growing, let alone the 50 to 60,000 in the fast growing. So what did we do? We started using this whole cell simulation methods that we talked about, this RDME. First, let me tell you quickly how you go about getting kinetic parameters for this. So the 5' to the 3' up there, that describes the rRNA on the small subunit. And one has known for many, many years that the assembly is hierarchical. Ever since the Japanese group of Nomura, that there are proteins that bind directly to the RNA, and then ones that bind to those, and then ones that bind to these. So it's hierarchical assembly with these ribosomal proteins. The trouble was about where to get the rate constants. And here comes the work of J.B. Williamson from Scripps. He did what are called pulse chase experiments. Whoops, excuse me. Went a little bit too fast. All right, here's pulse chase experiments. So let's say he added all of the proteins, the primary proteins, and then he would chase with a different isotope and then add in, say, the secondary proteins. And then he'd get from that a time course of how long it took those proteins to bind completely to the ribosome. We took his data, made an assembly map that you see over here, starting with the naked rRNA, and then you added these proteins, and the thickness of the lines just indicate how many of those simulations was it this primary protein, or this one, or this one, or this one, or this one that came first. And in these examples, done at about 30 degrees Celsius, it looks like S4 is really primarily nucleating the folding, and then the rest of them come in. And you see mostly red and yellow up here, because those are the ones attaching to the 5' and 3' and the central domain, so it sort of folds up the RNA from the 5' to the 3'. You can see that there's a bottleneck there. We also investigated that bottleneck using molecular dynamics, and I can refer you to that Biophysical Journal article if you're interested and see how we use the 2 techniques. Here you're starting to get that 3rd domain being formed, and then finally you get the intact small subunit. So now you have rates to make a model. It has 1,300 reactions, 251 species. I just told you about assembly. Now things can degrade. The messenger, we're going to assume everything. Messengers decay rather quickly, but they have been measured. And now we're talking about the messengers for the ribosomal proteins, right? And then you also have to worry about transcription, not only of the messengers for the ribosomal proteins, but you have to transcribe from the DNA. You have to get the 16S and the 23S. You got to get the rRNA as well. And then you have translation, so you got to read through the messengers, and you're going to assume an average rate of about 10 amino acids per second. There's a range of that from like 10 to 20. This is a pretty well accepted for these slow-growing E. coli. And then you have everything is going undergo diffusion, all the particles that are there. And they too have been measured by Johann Elf, one of those super resolution imaging guys, who gives you fantastic data. Here, just quickly, are the results. So here's a time course of 120 minutes, 2 hours. You're seeing you go from about 3,000 ribosomes to 6,000 ribosomes before it divides. And these little fluctuating values here of order of 10 each or those intermediates that was in that assembly map. And I want to remind you, I'm going to show you a movie of this, that you had circular DNA, and on that circular DNA, there were 7 operons for the rRNA, and roughly 5 for the ribosomal proteins, but only 1 copy of the ribosomal proteins. So let's look at this movie. Let's zoom into the nucleus, or the nucleoid region, and you'll see the rRNA operons. These are the operons for the ribosomal proteins. Messenger will be formed, it can diffuse, and then it will dock to the small subunit, and then it will assemble with the large subunit, and now you have an intact translating ribosome, and as you read through the messenger up here, it makes various proteins, and they get emitted here, and now you can start the assembly process once you have the 16S being formed from the rRNA operon. So there it is, and now you start the assembly, and one after the other, the proteins assemble to that, just like in that assembly map. It changes color to your back, and you now have the small subunit. Now imagine doing this again and again and again for 2 hours, and you can predict, whoop, I went too fast here, you can predict where the ribosomes will end up, and you should get something that's looking similar to Wolfram Baumeister's tomogram. All right, and then just look at it end on, and you see most of them are on the exterior or to the sides. Okay, so now everything I told you, that was 1 or 2 runs were done, but we also know there's another feature that we need to take into account, and that is correcting for the gene copies. Even though this is slow growing at the end, or halfway through that whole period of observation, you're having 2 copies of the DNA there. That means there should be 2 copies of every gene. We have the data from a colleague in our Physics Frontier Center who measured how many operons are there, and you can see it in the data that's here. And if that wasn't proof enough, people are beginning to use those single molecule, fish, so that's, again, STORM, measuring the mRNA distributions. And this is for a particular protein. This is a PTSG. That is the main sugar importer in bacteria. And so the work that was done by colleagues, Jinyi Fei, who's a postdoc of Taekyon Pa, who used to be at this university, had measured these distributions of the messengers. And we tried to fit it with a simple chemical master equation, ignoring initially the spatial distribution, and we could never get the full width of this correctly described until we took into account the fact that after a certain time, you have a second copy of the gene, and it too is making messengers. So if this is the division time of the cell, let's say it's 70 minutes, not 2 hours, and the gene you're looking at is here as, say, about 25 minutes, after 25 minutes, you're going to have a second copy of the gene. If one gene is making 10 copies, the other one will also be making 10, or you'll see a total of 20 in the cell after a certain period of transition, right? And you do have to take that into consideration if you're going to get the proper description of the messenger distributions as measured by STORM or any of these other super resolution imaging methods. And what was nice about this particular problem, we could formulate it as a master equation, the 2-state model, solve it, and use it also to compare it to our solutions, which we did here. And you see the mean, the variance, and the noise are defined by where that gene is sitting with respect to the doubling time, the cell doubling time, and the degradation rate of that messenger. Again, degradation rate of these messengers comes in. We tested it on a different set of mRNA data. This came from Rob Phillips in his Science article. So the histograms are his. Rob, too, knew that you had to account for gene duplication, so he fitted with 2 Gaussians here. But the red curve is ours, and it also agrees completely with the Gillespie solution of the chemical master equation that I showed you earlier. Okay? So, and then we went on and we did the messenger for the PTSG. it is not a case, what I told you about were cases of so-called constitutive expression. PTSG follows what we call regulated expression. So the gene can be off or on, then when it's on, it makes the messenger. So this you cannot solve analytically, but you could do an approximate model, which gives you a good starting point to figure out what K on and off should be, as well as the KT, right? And we did that, and there's the real data in the PNAS article, the histograms, and the green is our fit. And if you didn't know that it was regulated expression and tried to fit it with constitutive expression, that would be the red curve that you would get, right? So the precision of the experiments is now dictating that we have to really push our analytical models. Finally, to get a handle on the DNA doubling time, again, Tom Kuhlman went and labeled 14 different places along the circular DNA and looked when you went from having 1 copy to 2 copies of the gene. And he looked at it for a gene close to the origin, halfway here and then down here. And we had that information as a function of the length of the cell and the time. And from that, we could come up with a statistical model and get out of it the fact that for E. coli, this is not in general true for all the bacteria, the cell grows for a period of time before you start doubling the DNA. Then the DNA gets doubled, and it still takes a little bit of time, like 20 minutes, before you get the cells separated. All right? So we call those the B, the C, and the D periods. So we can take the previous model I showed you and implement it, and now we have a dividing cell, a slow-growing dividing cell. And if somebody, I showed this at a recent IPOLS meeting in Rice, I think it was Jose Onachek asked me, well, hey, the DNA didn't move its position within the next cell. That's true. We're not able at the moment to put in motions of the DNA in there. We're hoping that the models for DNA folding will become faster so that we'll be able to do that in the future. And not just to show you as a pretty movie, you can also get results of this dividing time. And again, you see the ribosomes located at the poles along the sides. What was interesting, this was a prediction of where you saw those intermediates in that assembly map. We said that should be in the center, in the nucleoid region. And from a recent talk I gave at Johns Hopkins, I was told, yes, indeed, they've been measured, and they are exactly, that is the primary position of it. Okay, so now, as I said, we've discussed the ribosome biogenesis. We've done simple model stochastic gene expression. And we want to move on to metabolism. And I think I was telling some of you, there is this report on progress in physics. That's the most recent paper that we have that explains all the networks and the different types of simulation methods that you can use. On just the minimal cell, we have one that we're revising for eLife about the metabolism, this section of the cell. Now, I'm sort of debating what to do with metabolism. There are special techniques. That's a field all into its own. In fact, it's one of the major components of systems biology. People try to write down all the reactions that are involved in metabolism and try to look at the flux that goes through them. And based on that, they can get an idea about the growth of the cell. So I'll try to do that very quickly, but by no means is it exhaustive. And I'm more than happy to talk to you guys about it. Or better yet, we'll give you the main reference paper. And this is from work of Bernard Paulson out at UCSD. So let's take a look at metabolism. So let's say you have a collection of cells. You took one of them out and you looked into that cell and you tried to write down all the reactions in metabolism. And I'm showing you sort of the key players. Like you take in glucose, it gets processed, the sugar gets broken down. It can be secreted as acetate or succinate. Some of it can be funneled out through pyruvate into a so-called TCA cycle where you make other amino acids. You can also have it go over through the pentose pathway and make nucleotides, or you can have it go siphon part of it off here after just breaking down the sugar a bit and start making the lipid. But the metabolic maps, they try to write down all the reactions that you have, show you everything that the cell can take up, like glucose, oxygen, secrete. And in the case of E. coli, you can also take up acetate. As I said, this is the leader in this field, and he has the most exhaustive metabolic maps for E. coli that are out there. So how do you study these, right? Typically, they study them at steady state. You look at the cell growth and sort of look at it in mid-exponential state and sort of assume that the metabolites are not changing substantially in that and come up with the flux through each of those reactions and can relate that to a growth. So let me get out of this for a second and get into this PowerPoint that I typically use for flux balance analysis. So again, what are the steps? First, you do a genome scale metabolic reconstruction in that you try to identify as many of the reactions involved in metabolism as possible. You make that map up there, which I just showed you. You write down all those reactions, right? Like, and this is about the metabolites, A, B, C, D, and reactions that convert them from one metabolite into another metabolite. And so let's say you have N reactions, you put those here, plus you put in the ones about the uptake and the secretion, plus you have a term that's called the biomass equation. And that just says, okay, I take up glucose and all these components, I've weighed the cell, I know I have to create this much biomass if the cell is to grow, right? So that's an equation that has all the building blocks in it. And what you're looking for is the flux through each of these reactions. And you want it at steady state, so instead of looking at the time variation, you want to look at it when it no longer changes, or you have this easy system of homogeneous equations to solve. Trouble is, as you notice, more equations than metabolites, or the other way around, depending what system you're looking at. So you won't necessarily have a unique solution unless you start imposing constraints. So what are the typical constraints that you impose on that, solving that stoichiometric matrix times the flux matrix? The more and more constraints you put, the more you get towards optimal solutions. And even there, it's optimal in that it has minimal flux through, say, each of the reactions. That's called parsimonious solution. But what are the typical constraints? Well, if you have single molecule measurements, maybe you have microfluidics, maybe you have proteomics, put that in there. Do I know something about the RNA-seq? You might. And then you might be so bold as to try and infer protein from amounts from RNA-seq information. Cautiously, but you can do it. So there are other constraints that you can use. Most of the time, people use proteomics constraints. So again, here's like a more complicated eukaryotic cell. You have things coming in, you have reactions taking place, you have secretion. And now if you look at that biomass, what it really is telling you, getting these building blocks, making the DNA, making the ribosomes, making the membrane to have the cell to grow. And so if you go over here and try to solve this set of equations, subject to getting a max, what you're trying to optimize is having this equation be maximized for growth. You can try to put constraints on these or bounds on these with any information that you have. And typically what you'll do is if you know something about that reaction, like you know it's KCAT, and you know from proteomics what the mean protein copy number is, you could put that in there as a constraint when you're solving this equation. Now the KCATs, I talked to you briefly about that. Where do you get them? Out of this Braunschweig-enzymatische Datenbase, Brenda. You can just go look it up, and it gives you the KMs and the turnover numbers here. And this is for this is this first step in taking in glucose and converting it to alpha-D-glucose 6-phosphate. Right, so that's there, you can put those in. And now let's go take a look at Sunny Shee's experiment. That was this middle panel here. He, as I said, he labeled like 1,000 proteins, put it through a microfluidic device, and measured the protein distribution. Say this was for adenylase, and this was for the alpha subunit of ATP synthase. So he has a distribution. Some cells have more, some cells have less of that particular protein in it. So what you can do is you use that information, put your bounds on it. You know something about the exchange reactions. What is the average uptake rate of glucose in a cell? What is the average oxygen uptake? And you can put that into the equation, try to solve it to get the maximum growth rate, and you do that, then you will get, well, let's put it this way. What you will do, you will sample out of this distribution. So you're going to have a different copy number for every sample. So let's say you sample 100,000 times. So you're going to have really 100,000 different populations. You'll do that and solve for each one of them for their growth rate, and then you'll get a distribution of growth rates. And here I did 1 million samples. The line that you see here is the average optical density, that when they hold it up and say, the average growth rate is 0.34 per hour, that's what they're recording. But you see that there's some cells that are growing faster and some cells that are growing more slowly. And what you can do computationally is go in and examine those cells. What's different about the protein population here than here, and what part of that metabolic map is being used more or less? So if you start looking as a function of the growth rate, and I just wanted to look at the main pathway, glycolysis, and you see that there are cells, and each dot here corresponds to one of those different cells that tend to be not being used very much up here, and there's others more so here, and then there's a whole bunch that are using the so-called ED pathway. So let's go take a look at glycolysis. That's this main pathway here. This would be taking in glucose through that PTSG and having it just being broken down like you see in your textbooks. There is a shunt here called the ED pathway that goes this way. And if you notice, as I said, I don't have much time to explain these things. I'm trying to show you the metabolites, but the metabolites are being converted from one to the other through enzymes. You'll see that there's very little markings on here. So you have very few enzymes that are used here compared to here. So you're able to grow faster, but you can, you're not having to use so many proteins. So there's a balance between proteins and growth rate, the amount that you use. So that was one example that we saw. We can also look what happened to those slow-growing guys. And it turned out a lot of them that were slow-growing were secreting acetate. And again, we could go over and look at our metabolic map, have now the thickness of the lines indicate the flux. So here they are. There's a lot of flux through the main glycolysis pathway. You're getting secretion of the acetate. You're not getting so much into the TCA, and that would also lead to CO2 production. But now, when you go to those high rate of growth, all of a sudden, it's not producing acetate anymore. It's using this part of the cycle, and now I think this is the succinate, a formate that's being secreted. So that's how you, doing a theory, can go and make predictions. This has since been validated by a group at the Weizmann Institute, but what was more interesting, when we published this growth distribution, and there are only like 3 or 4 of them that are in the literature. We have one, and there's another one that was done for yeast. Oh boy, too fast. That was done here for yeast, and the guy wrote us, that was Siegel, he said, why does my distribution look like your distribution for E. coli? And I go, good question. So unfortunately, to answer that took us over a year to do, and here was our solution, was then published in 2017. So that is our fit to his data. And the reason it's complicated is that glycolysis is not just in the cytoplasm. Parts of it, like that TCA cycle, is over here in the mitochondria. So you have to look at what happens to the pyruvate, how much of it gets secreted, how much of it goes into ethanol. We were able to do this using a metabolic map there's a community of people working on it. We improved the last version of it, and we could get pretty good agreement to the carbon-13 measurements that were done on the main glycolysis pathway. So the fluxes agreed fairly well with that. So that's the type of thing that you can do if you have really good metabolic maps. You can try to be predictive. But having said that even E. coli has like 1,800 genes missing, there might be some experimental condition where this won't give you such great agreement. So just keep it in the back of your mind. There might be reactions you don't know anything about yet that you might have to put in there. Because people tend to work under just a few different media. And that enables us to compare and use each other's data, but that doesn't really allow you to probe the vast array of conditions that could be out there. Okay, so now let me get out of this and return to the world of metabolism. And now what we want to do is not just look at one cell, let's think about it in a colony. And how are these guys that are on the periphery and maybe in the center different from the ones that are perhaps in direct contact with the food? So again, we have the same metabolic map in each case, but what we're going to look at now is we're going to put a single cell and watch it grow on an agar that has this amount of glucose and trace 9 metals, and M9 is just telling you about the kind of salts that we have in there. And now we'll have a sublattice, we'll have lattice or subvolumes across the colony, but now it's not over just a single cell. In any subvolume, there can be multiple E. coli cells. And what we're interested in is really solving this reaction fusion equation, and along with the reactions going on inside of the cell. So we're interested in, okay, the stuff that gets secreted out of these cells here, like acetate or uptake from oxygen, how is that affecting the local environment of those cells? How does that affect what they take up? Do I see more or less glucose? Does that make me secrete more or less acetate? And how about the CO2, or what about oxygen, right? So we can use simple arguments to look at the uptake in one of those subvolumes of those average cells that are there. We can go solve this problem, steady state, and then move on and allow the distributions to change and do it again, and you keep cycling through. And that growth rate that you get, all that does is gives me a parameter to tell me if the cells are growing, when do I start pushing them into the neighboring subvolumes? It's a single parameter I get out of this. It's a very simple treatment to get the growth proper. And you're, again, just going to assume that the cell mass is exponentially, increasing there. And now here's a little movie of the cell starting off with a single cell growing into a colony. And now I've attached to different strata in the colony what part of the metabolic network is active, right? And to validate that, we did a couple things. We put reporters on, say, up here where the glucose utilization takes place, or one down here where you have acetate utilization. And so you can look over here. We can also measure the colony height, the diameter and its height, compare it to the experiments here. And the wiggly lines here just means when we had that growth factor and we moved cells into neighboring subvolumes, did we assume that the volume was half filled, or did it have to be 80% filled before we push cells out? And you can see, I think, about 80% is probably a little bit better fit for it. That would be the red curve, but 6.5 is also fine. We also looked at the oxygen distribution as well as the glucose distribution. Of course, you should never see glucose here in the air above the colony, but you can see a change here and its variation within the colony. So after a while, the guys at the top are not seeing much glucose. And in fact, we validated this doing a set of experiments. So we put in a plasmid with two reporters, one for glucose utilization and one for acetate utilization. And we predicted after 30 to 40 hours, the cells at the top, if you shine light on them, should glow green. And the ones at the bottom should be red. And the ones at the bottom, they're going to be taking up the glucose, producing acetate, and the ones at the top are going to be consuming it. And here's the actual experiment from the structured illumination when we cut through the colony far away from the agar, so you should see only green. And when you went down here and did a cut through cells close to the agar, you should only see red for the glucose utilization. And I don't know if you can see this, but on the side is a cut through the colony. And you can see it looks a lot like this. So this was one of the nicer validations of a cell simulation. But it's not perfect. If I were to tell you comparison of the intensities, it doesn't quite agree. But it's still strong enough signal that you are seeing the variation in the utilization of glucose and acetate. Since I know some of you are from Rice and they're interested in the physics of the metabolism of cancer, this cross-feeding that we see of the acetate being produced down here and consumed up here is a fact that they're using in eukaryotic cells. Here's the blood vessel, and if you have a hypoxic cell, it's going to be consuming glucose and producing lactate. And one of the strategies that this fellow came up with, and he now has a patent on it, is he tries to block the use of these other cells of the lactate, and that then slows the growth of the tumor. So this, if you look under lactate cross-feeding, you'll find this fellow's patent. Right, so it can be extremely helpful in trying to understand these things. Okay. So what I want to address next is now the metabolism of this minimal cell, because as I promised, it's going to be much simpler than the one of E. coli, and then this will hopefully give us a really good overview of the principles of life. By that I'm meaning the energy requirements as well as the fundamental reactions that you have to have. This becomes interesting because there's a real push going on in the scientific community to work on either synthetic cells or artificial cells. And people keep on trying to define, well, what does it mean to be a cell? What properties does it have to have? And there's even a German consortium where they don't put DNA in it. They go, well, if you have like 4 principles that have to be maintained, a cell has to be able to reproduce, it has to have a membrane that can bring in and secrete things, it has to have DNA, you have to get robust reproducible sizes and reproduction in the cell. They go, oh, there's 4 basic tenets, but I'm only going to try to satisfy 2 of them at any one time. So, like, there was a joke in this one workshop that we organized about, hey, let's just make the cell membrane out of stainless steel and drill holes in it. And somebody yells, oh, you mean like a battery or something? And so if you really push it to the extremes, you start seeing what people are, how they're struggling to understand what it means to be alive, right? But it is ongoing. There's a lot of engineering processes that people are looking. If I take some of these basic reactions and put them in liposomes or in artificial membranes, can I use as a factory to make something? So there's a whole community of engineers that are pushing that aspect. All right, so let's look at the minimal cell. As I said, it came out of a science article. That was just SYN3. The one that we've submitted with them is SYN3A because they had to add back in a few genes into this to get consistent sizes. This is 400 nanometers across. These are the 2 lead scientists. One is Clyde Hutchinson and the other one is Hamilton Smith. He's a former University of Illinois graduate. He studied mathematics here and went to Berkeley. And when he decided he wasn't going to get the Field Prize in mathematics, he switched to biology and got the Nobel Prize for restriction enzymes. So these are great Skype sessions every 2 weeks with these guys, and very thoughtful planning and validating not only their experiments but also our modeling. So that is the postdoc in my group who has built the metabolic model. It's made up of 155 genes out of those 493. What is interesting is this part down here involving ribosome biogenesis and genetic information processing. It's pretty much the same as what we had in E. coli. A few differences, but first approximation, you can carry it over. And now here is the metabolic map on a single page. This cell, you have to feed in everything. Like to make a nucleotide, you have to feed in a nucleobase or a nucleoside, and in 2 or 3 steps, you're at the pyrimidines, or the purines, or the pyrimidines. The same way with the amino acids, you have to bring in the amino acids or read in a di or a tripeptide, break it apart to get the amino acids. For the lipids, you have to feed in fatty acids, even glycerol. So this can be like chains of C16 you have to bring into these. Here's the glycolysis pathway. That's pretty much the same as you see in E. coli, but the connections between it and nucleotide metabolism, lipid metabolism, amino acid metabolism, there is no CCA cycle, for example, of this, or much smaller. The other thing I want to point out is the energy requirements are so much different. To make a nucleotide takes 50 ATP in E. coli. This takes 3 to do it. So I think that's why it's still able to grow in 2 hours, provided you give it a rich environment to live in. And we're just now trying to figure out what sort of is the minimal medium that we can use to make this thing grow at the same rate. Just to give you a quick breakdown of those processes that we talked about. So as I said, there's cell growth, there's transcription, translation of the genetic information processing, metabolism, we discussed that, and again, those 91 genes that we don't know, they're in these category off by itself. There's another column that we've added here for essentiality, and I think I told you that they got that from trying to insert transposons into the various genes. And if you don't see tick marks here, like this gene, they don't know what it is, but it's essential, because you really couldn't add much into it. Some of them, it looks like there's some variation. Maybe yes, maybe no. This is looking at round 1 or round 4 of this study. And it turns out these are often multiple transporters of, say, dipeptides. So maybe you can knock out some of them, but not all of them. And you can summarize everything that you've done in terms of, again, these gene maps. But now let's go look at this side. It's no longer genes. I'm using the proteomics data we have for this. So the polygons at different sizes. And what's going to be interesting is, let's go up here and look at the non-essential. There's a lot of that gray is there. But more interesting down here for essential, there's still some of those 91 proteins, that's about 30 of them that are essential. We just don't know what they do yet, right? So, and they're there. Some of them are in small quantities. Others look like they're comparable to the distributions that, or the amounts that you see in the proteomic studies of other systems. And just to give you a zoom in of one of those metabolic networks, here's where I was telling you where you read in either the base or the nucleoside, and then in a few steps you're down here to ATP. The same way over here for guanine, you're down here to GTP. And the numbers here indicate they know what genes they are. And so one's trying to do experiments where you can vary base or nucleosides. and figure out what is exactly happening here. One of the things I myself found very interesting to learn from this was the mechanisms of DNA repair. Many of them are removed here. And it is a real problem to keep UTP out of DNA, right? You have both the deoxy version here and the RNA version here, but you really don't want TUTP. And there are just very few mechanisms that help you to do this, and it wasn't clear what was going on in that system. So that's one of the things that we're still looking at. We think we've identified the genes that help get rid of DUTP so that you don't slowly degrade the DNA. And if you want to have a zoomed in picture for the lipid metabolism, here you're coming with the fatty acids, and boom, in a few steps you're down here to cardiolipids, right? And finally, with the amino acids, you can read in either the amino acids here, those are some of those redundant operators, or you can read in a peptide. Oh, excuse me. These are the permeases to read in the amino acids. Here are the peptides. And then you break them down, and then that feeds into the charging of the tRNA. Now, as I said, we don't know much about the system in terms of, we don't have a tomogram yet for it. That is the cell. That's how it looks like in our software. People say it looks like a Minecraft game, and we wish we could turn it into one and make lots of money. So, you know, one possibility is maybe the DNA is all located or, you know, condensed and in the center of the cell, and the ribosomes are on the outside, diffusing along. So that would be a picture where you have the DNA. There's some of the other proteins that are involved in the reaction to fusion, the description, and then you have the ribosomes diffusing. This presents another challenge in how to do those simulations where you're dealing not only with different concentrations, different size particles, and your proper choice of your reaction subvolumes. And finally, just to summarize this, as I said, the minimal cell and any cell that you would do will address all these issues of concentrations. Like DNA, you're in nanomolar regions. You get into the amino acids, ATP, you're over here in the millimolar, and you have to be able to mix and test these different methodologies and how do they communicate information from one description to the next. So we think we have a good handle how to do this. So we are trying to build the entire model for the minimal cell, combining all the networks together. It's ongoing work as I speak. And the idea would be to compare it to experiments as well as, say, molecular dynamics simulations, because having NAMD in our NIH center, they've been working to expand that program to be able to do 2.5 billion atom simulations. So we could build a state of the cell, we could put back in, just like when we did with the ribosomes, the structures, and see if it's a physically acceptable state of a cell or does it blow the whole thing apart. And this is going to take at least 4 to 5 years because of the fact that so many of the things that are in these crevices are transporters. So this is just an idealized picture of its membrane where we put in a 1,000 aquaporins because we have the structures available for those. And for those of you who never met my husband, as I said, he was the leader of this, and this was a friendly bet between us that he would get it into NAMD, I would get it done through lattice microbes, and we would compare results for a microsecond. All right, so now let's turn our attention to, well, maybe before I get to the program, are there any questions about what we just talked about, metabolism and the genetic information processing? Anything that you guys, general questions that you have on that? Anybody? I know I'm going rather fast, but I just realized I only got through about a quarter of the material in the first hour, so put it into high gear, so to say. No? Okay, so now we're getting into the hands-on sections and more about the programs. And as I said, we've already introduced to you Mike Halleck and Tyler Ernst. So as I said, Mike is sort of our gatekeeper of releasing the lattice microbes so that it obeys good software engineering practices. And because we believe so strongly in the collaboration with the experimentalists, we do that through these Python Jupyter notebooks. And that's what you'll learn about. We'll also use it to input data into the cell simulations and examine them. You can also use VMD to examine the simulations. And that one movie I showed you on ribosome biogenesis, for those of you who know graphic software, that was a Blender movie. I don't know if anybody's made a Blender movie in their lives. It's pretty cool software, but there is a overhead to get into it, right? So as I said, by doing this, working with the experimentalists, we can agree on what sort of fluorescence cutoffs we want to use. Then we set up the cell simulations. We can run it on the supercomputers, on a GPU cluster, or you guys will be running it on the Amazon web. And then these are sort of some of the projects that we've looked at: eukaryotic cells, E. coli, individual cells, as well as colonies of E. coli cells. So we've talked primarily about bacterial cells. Now I want to turn our attention to eukaryotic cells. What's the main difference? As I said, you will have one of the leading experimentalists here on Friday to present to you how they did the experiments on the stem cells. And this picture here summarizes work that, again, was done with Wolfgang Baumeister. He published his pictures. This is, I think the title of the paper was The Molecular Sociology at the Periphery of a HeLa Cell. And we published an article together in the Festschrift to my husband where we were showing what happens with their tomography data. So this is real 3D data set from his microscopes there in Martin's Reed. Then they can segment the tomograms. So from that picture and from some of those data here, do template matching and see that's a nuclear pore complex. Those are ribosomes, and in fact, I can see that that's the large and the small subunit. And these are microtubules that are come here. So you get really a beautiful view. This is coming in through, being shown to you through VMD. But now when you put it into our cell simulations, we sort of blur it because we have a certain resolution to our subvolumes, right? So it's almost, feel sorry, beautiful. They've segmented, really increased the resolution here, and then we go and blur it again. But that's what goes into the cell simulations. And to show it to you even more close, better, again, the top part of this picture is after they've done segmentation and fitting. Thrown in a few more features of the cell, namely the endoplasmal reticulum, and we have some of this of the actin and the microtubules. And here you see very clearly the nuclear pore complexes that allow the DNA's in the nucleus, the messengers are made, and they come out through those nuclear pores and get translated on these ribosomes that are sitting outside of the nucleus. So again, they've color-coded them with the large and the small subunit. Again, zoom in on just one of the nuclear pores, and that's how it would appear in the cell simulations. But like when Zala did her simulations on HeLa cells, she was taking her diameters and her sizes of these nuclear pore complexes directly from the tomograms. Quite often with the tomograms, you do not have the complete cell. You only have a cut through it. Sometimes they put several cuts together, but invariably for the bigger cells, you only have slices through it. So you have to take information like this plus other lower resolution information to sort of build up the entire cell. Okay. And this was a I won't bother you with this. There's Zala again, and if you have further questions, she can tell you how she used LM and PyLM sort of to build up her HeLa cell, how she added reactions and diffusions, and then she added in the particles. So this is pretty much the workflow. You start with building a geometry. And if you have tomogram data, that's wonderful. If you just want to try out something, then also Tyler will show you how to do that, how to build a cell from scratch. But it's great if you have as much realistic information as possible, but there's also a value in just playing with things, changing the sizes of them. All right. When you're building up these complex eukaryotic cells, which have a cell nucleus, mitochondria, ER, you have to sort of use, at least if you're using PyLM, use a more solid geometry approach. And there is an online manual for this of these calls to be using. If you want to make things a sphere or a hemisphere or a cone or a capsule if you're doing a E. coli. So we have some basic shapes in there for you to work with. And then you have to start specifying what are the regions. Like in her HeLa cell model, she has 11 sites. She has the extracellular, the plasma membrane, the cytoplasm, the inside of the nucleus. She is also looking at particular Cajal bodies and nuclear speckles because they seem to be important. And for those of you who don't know it, it's a rather new into the literature. They see that inside of the nucleus looks like there can be temporary formation of organelles that don't have membranes. They come and they go. And sometimes it's like you're storing stuff there for, say, the spliceosome or for ribosome biogenesis. They can be there under certain conditions and disappear or melt into one another in other conditions. And then she has the ER, the mitochondria, the nuclear pore complexes. And then she started adding species. Like here, she wanted to look at just 200 genes inside of the nucleus. So her picture then of the HeLa cell would be this. So you're seeing that this is the nucleus. You're seeing the speckles, the Cajal bodies. These irregular shapes are the ER, and then one can add the that is the ER, and then you add the mitochondria into there. All right, so then you have the cell built. What about something smaller? The HeLa cell is at least 10, maybe 50 times larger than a yeast cell. So yeast is the simplest eukaryotic system that you can do. So this is looking down on a yeast cell. This is looking into the nucleus. You can see the cell wall out here. So this nucleus is sitting sort of shoved off to one side of the cell. You're seeing the ribosomes. You're seeing a mitochondria here, and here's a little bit of the endoplasmic reticulum. That's the brown. And now let's turn that slice on its side, and you can see the nuclear pores. You can see the ribosomes. And you do see some structure. You do see some of the DNA into the cell nucleus, but it's not really enough to see the folding of it. They're improving on that, so that is going to get better and better. There's also additional labeling on top of the tomograms. And Tyler will be able to show you how you start with this information and build up an entire yeast cell. This was like one rendition that we built, and we wanted to just consider the cell surrounded by a certain amount of sugar. Like in this case, it was micromolar, not millimolars. And we wanted to look at flow in through these transporters, the green dots, and then go in and turn off a gene here. It was supposed to be just a generic switch, and we looked at it as a function of time, how long it took things to go from the outside to the inside of the cell. So this was just very generic. What is a better study is what we did next, which is to look at a specific switch in yeast. It's called the galactose switch. And the reason we did that is there's beautiful single molecule studies have been done on it long ago by Uden Arden. There's also the kinetic networks have been written, and down and tested, so we could use those. And what goes on here is you have extracellular galactose in millimolar concentrations. It comes in through these transporters. They're called G2. And this, what happens to that galactose? Well, it can be metabolized, or it can go and stick to a transcription factor, which is suppressing 4 genes here. The more it binds to this, the less likely it is to go into the cell nucleus, and then these genes turn on, and you have feedback loops here. So what you see, if you were just to look at the transcription factor that's repressing these genes, is if you start pouring in sugar, is that the amount of the transcription factor goes up in the cytoplasm and goes down in the cell nucleus. What about the intercellular galactose concentration? Well, you started off with 0, and then it starts going slowly up. It'll never get up to 11 millimolar. Here it's like 40% of that, say roughly, like, let's say 4 millimolar, because it's being metabolized in this case. And so that's in high concentrations. So what we're going to have to do is to get the stochastic gene expression described accurately. We're going to use a combination of the CME, the Gillespie algorithm for the reactions, but communicate some of the reactions that are involving directly all those millimolars of galactose through simple ODEs, and we'll trade information back at certain communication time steps. Now, let me show you first the results of these runs in terms of how long can we simulate yeast in a day. Well, we can get 1.5 hours of yeast in a day, but you better use one of the better GPUs that's out there. But for those of you who are computational scientists, this is the class of GPUs that's going to be available on the newest supercomputer that's being built at Oak Ridge, right? And so we're talking Titans and Voltas here that you're going to have to be able to do this study. And I think I never showed you these times. You don't get quite as good performance in doing with yeast, with E. coli. These are on different GPUs. So sometimes the bigger the system, the better you can spread it across the nodes, and you can even get higher increase in performance in doing those. So let me now go one step more and show you the oh, I hope this works, the movie of that galactose switch. So we're going to zoom in on the cell nucleus. Here's the gene for the transporter, G2, that's going to let the galactose in. When it's off, it's red. When it's on, it'll turn green. And then when it's being translated in one of the 180,000 ribosomes, it'll be orange. And then it makes this membrane protein, which are the cyan colors, and those will diffuse and insert into the cell membrane. And because the nucleus is sitting asymmetrically in the cell, and these holes here are the nuclear pore complexes, you'll see more of the permeases forming over here, right? And that's about, that was 59 minutes, that simulation. And it was an RDME using CME in the reactions and the subvolumes, and the ODEs for part of the for the reactions involving lots of the galactose. Now, I seem to have just some reason or another, I took that part of it out. I thought I might show you quickly. Might find them. If not, it's okay. Instead, what I can do is go here and then just show you all the people that are involved in developing these techniques. So up the top here are some of my experimental colleagues. Tay Kip and Tom Pullman are from our Physics Frontier Center. It's an NSF Center for the Physics of Living Cells. Some of these others, including Martin Grubela, which I don't have his picture here, and Sarah Woodson's at Johns Hopkins. And then the people providing the tomograms are from the Max Planck, that's Wolfgang Baumeister. One of his main disciples, Elizabeth Vila, is now at UCSD, and she's doing tomograms not only of the yeast, but of the minimal cell. And then these are the colleagues from the Greg Venter Institute. So Marion, he's with our Physics Frontier Center. This is Greg Venter and Hamilton Smith and Clyde, they're involved in there. And then the colleagues that are from our NIH Center, like John Stone for his help with the visualization, that's him again. And these are some of the former students like Joe Peterson and John Cole and Pioche Lapsapar, who worked on the metabolic modeling of the metabolic models, both for E. coli, for yeast, and particularly Joe for his work on the methanogens, which I didn't even talk about because I think there's very few people who work on methanogens besides people with DOE grants, because they're ones that are important for DOE because of the methane production. They grow under extreme conditions, so NASA likes them. They think they may be producing methane on Mars. So that's a whole different set of people that are interested in those reactions. And it's really different, is that it doesn't have a glycolysis pathway. It doesn't even take up glucose. They tried to build in a glucose transporter. It didn't help. You know, what does it take in? It takes in methanol, methylamines. It makes methane. It can make CO2. So it's very important for that balance of methane and CO2. And if you go to swamps and you smell the methane, that's those guys. If you go over to rice paddies or cow farms, those methanogens are growing like crazy there. And they've now got an importance even for NIH and not just DOE, because they're finding them, they're in your gut. And if you don't have the right balance of them, it can lead to obesity. So there's now people looking and doing NIH research on these methanogens. And then, of course, there's Mike, you've already seen, and Tyler, and David, and here's Zala. And this was Jinji Fei, when we did the work with her on the importance of gene duplication and getting the storm measurements accurately explained for messenger distributions. She was a postdoc of Tay Kip Ha, and now she's faculty at the University of Chicago. And many of the people who have worked on these problems before are also professors. So like I told you about Elijah, he's at Johns Hopkins, and there was another physics postdoc, he's a professor at the Hebrew University in Jerusalem, and Lars Kohler, he worked on the beautiful cell simulations on the colony, and he's now at Argonne National Labs. So, boy, when you speak fast, you can cover a ton of slides. So, sort of done with the main part of the lecture. I think now we're supposed to have some question and answer periods, and then what, to 12:30? And then we have lunch back upstairs. So I think you've seen a little bit of everything that you're going to learn about. And we'll start up right after lunch by Mike. You'll be giving them the information to get onto the cloud. Is that correct?
Right.
Okay. And he'll give you a lecture. And after that, we'll have Marcelo start explaining the Jupyter notebooks. So, and as I said, it's really important that you spend time learning how to use these Jupyter notebooks. And then tomorrow to go through the lattice microbes tutorials. I've even asked Marcelo to show you the first of the tutorials where we just take the Michaelis-Menten equation, I think, and solve, or maybe even a bimolecular reaction, and solve it using just ordinary differential equations and comparing it to doing it stochastically. Did you put that one in?
No, you can do that.
Yeah, okay, you did do that. But he will do that.
It's all in there.
Okay, so are there any questions right now that I can answer before we go into the lunch? Any? Everything's perfectly clear. Okay, the other thing was we were trying to leave Friday afternoon. If you have particular problems that you're working on right now or want to work on, then hopefully try to take the time this afternoon and also on Wednesday to talk with us. We can try to work with you to help set up a cell simulation. If you think that would be useful. And so that you can spend Wednesday and Friday afternoon doing the advanced tutorials, which is more onto the eukaryotic systems. So, or you can work on your own system. This is what we typically do with these other workshops. We encourage people to bring a problem that they're interested in, and we'll help you set it up so you can take it home and work on it there. And if you have questions, you'll know who, you've met enough people here, you know with whom you can communicate to do this, okay? So if you need also some input for your model that's in your lab, well, you've got 2 days to get it transferred over to here. And you should be able to get on with guest accounts to the, how do they do that? Is the guest account for Beckman here? Or is it the guest account for the UIUC? You know what you use, Mike?
Should we let one of that guests people not already choose that network and there's a small.
Okay, okay. Also, EDU Roam works, but I don't know how, I know with LMU it sucked half the time. But sometimes it was better than the biophysics link that they had there in the Applied Physics Institute. But so do you guys from Germany use the EDU Roam?
Yeah,
Yeah, try that. That should work fine.
Yeah.
Provided you're, you know.
For you? It doesn't work for you?
Well, maybe your password. I had trouble when I was over there because I had just updated my password. And they, I don't know about your place, but they renewed, you have to renew it every once in a while. Right? Okay. Anything else? Any other pressing questions? Nope. Okay. So, as I said, I think, I don't know where all of you are staying. We can also discuss in the afternoon how you get to where the social dinner is tonight. And maybe if there are no further questions, I'm just trying to find where I have this one thing I wanted to show you on the yeast simulations. Gave so many talks recently, I'm trying to figure out where I did this. Here. Oh yeah, here we are. So let me start with this. Our yeast model. And again, this is yet another view of that tomogram. You can just sort of see the outline of the cell nucleus there, right? Here's the cell wall, and then this would be how Tyler would start building up this tomogram to get to a full cell. So you take stripe after stripe, you have the nucleus, and then the rest of it, and then voila, you have an entire cell. Okay? So that's step one, to build up the cell. And then just to remind you of the characteristic scales that are in these problems. So let's say the yeast is about 4 microns across here. And let's look at the concentrations, the diffusion coefficients, and the diffusion times of various components. So if you're a sugar, you're in thousands of micromolars, or essentially millimolar, right? You have really fast diffusion coefficients. Diffusion time is it's really short. You look at proteins, it's about 1,000 times um, lower in concentration. The fusion coefficient is several, at least 2 orders of magnitude less, 5 versus 200. And even at the gene, the DNA, its motion is, I don't know, we should ask Vihela, if you've looked enough at the folding, if the motion of any one gene, say in the eukaryotic DNA, kinds of motion you anticipate, is it going to have a diffusion coefficient about 10 to the minus 5 square microns per second?
Yeah, I was about to ask you about that because so this diffusion coefficient that you give, they are hydrodynamics or measured?
Hey, Tyler, were these coming in from bio numbers? This diffusion coefficient for the DNA in yeast?
I think I just chose.
I mean, what you can do is you know, get an average, like if this cell has to duplicate, how much can the DNA move within an hour and a half? So you might be able to translate that. But there have been some attempts to label, like if you use a DAPI, and then you can, or even zoom in on one particular gene and sort of see it, you get a rough idea of its diffusion coefficients. But this is one of the things that we would love to have better estimates of this.
We actually had just a paper that came out last week
Okay.
on this specific issue. It's not a simple thing because the thing is that if you assume there's a sphere of a certain size, mass, and it moves at a certain temperature, you get certain diffusion coefficients, right?
Okay, so
Then-
let's translate. If you assume the cell is a sphere, but what are you assuming on the DNA?
No, the DNA. Let's
The
say
DNA,
that
okay.
The DNA is some kind of renormalized sphere
Okay,
that
okay,
moves at a certain temperature.
right.
Then you measure some diffusion. Then you can calculate using Stochastic some diffusion coefficient, right?
So one is trying to do that through hydrodynamic arguments, but that's
Yes,
only a
so-
section at a time.
Yes.
Okay.
So we started from there. Of course, what you measure in the cell is completely different. So they measure that it's subdiffusive, it's viscoelastic, and they measure that it's coherent. So in the sense that if a piece of DNA moves in this direction here, this tends to move in the same direction. So it's another complicated thing that is a combined effect of the fact that there's a cellular matrix inside, and there's other problems, there's the tension binding. And the whole thing has been explained in a lot of difficult and different
Right.
theoretical models. We found out that with our model, we get roughly correct behavior from the model, starting from the diffusion data hydrodynamics, right? So but we start from the hydrodynamics, and then we add all the interactions, so we get this subdiffusing viscoelastic behavior. In terms of the number, we don't get it right, but that's not surprising because.
And this is on the human cell that you're doing this on, the human chromosome?
So in this paper we added a few papers. So most of the data are from a couple of different groups. A group in Israel, Barkai and Garini.
But what's the organism?
And then they worked mostly on the bacteria. But I think there's some data also on HeLa and on human, more
Right,
from this.
right.
But if you look at the paper, I find it.
Because it doesn't, it's getting more complicated. This is, of course, for a eukaryotic cell for an estimate on yeast, because when you have a gene being on, there seems to be growing evidence that that piece of the DNA either moves towards one of the nuclear pores, or has an interaction with the speckles. So there is motion in one phase of the cell cycle, and then that's totally different from the one where you're having the spindles pulling it apart and to form the cell duplication. But just within a certain, like say the S phase, how much motion are you seeing of the DNA? So did you try to do this in a certain phase of the cell cycle?
This is all ensemble. So
Okay.
all these directed movement cannot be captured by
Right.
our model. But
Right.
yeah, I know. Belmont is seeing a lot of this
Right.
directed motion, but
So
sometimes
this is a real challenging aspect to all the simulations. And I would even say, even into DNA, almost everybody uses DNA chromosome capture data to try to fold the DNA, but then, you know, in E. coli, you have different proteins interacting with the DNA than you do, of course, in the eukaryotic system. And all you know is I've got to have one copy here, and towards the end of the cell cycle, I have to get the second copy migrating over here. And again, there's some work from the Israeli groups that have used labeled nucleotides that you could follow the growing strand. So that data looks to be pretty good, and if you were able to put that data, that would be excellent. But that's like 5 years old, and maybe it's something more recent was done. But the problem, as I said, I just want to point out the challenges. They're not insurmountable, but it did cause us to stop and develop hybrid methods because the concentrations and the time scales, they don't overlap. If you're asking about sugars, proteins, DNA, different time scales for that. And now let's this is the picture I was looking for, is implying one of those hybrid methods. So now here you have the transporter, the G2, bringing in the galactose. It can be metabolized here, or it can interact now with the transcription factor G80 and prevent it from getting into the cell, which then allows more of these 4 genes to be made, and one of them is the transporter. So you'll go from just having a few here to having them all around the cell membrane. And within each of those volumes, we will do the chemical master equation. But for these guys, where you were having the millimolar concentrations of the galactose, you're going to treat that by a differential equation solver. That's the particular name. It is called L-SODA. It's one of the oldest ones. But I'm telling you, and you can use these in the Python notebook. Turns out this one, I think it's in SciPy, isn't that right, Marcelo? L-SODA?
Yes, it is.
Okay. Yeah, David would know too. And part of the problem was one of our reviewers asked us, well, you know, you had these, we tried in the paper to come up with a good optimal communication time. You know, as long as you communicated more often than this, you would capture all the fluctuations that are known to exist in the cell. And then he said, well, as an extreme case, call the differential equation solver every time you have a stochastic event. And somehow things just didn't look right, and then we realized it was a problem with L-SODA. It's because if you call it a differential equation solver and give it a time that is too rapid, it just gives you back your initial condition, but doesn't tell you, which is bad, right? So it may be a classical and one of the most highly valued methods, but it sucks in that it doesn't give you all these sufficient error warnings. And what happens as you move forward with computational work, some of these established computational packages, you know, people didn't worry about fluctuations. They were happy just to use ODEs and PDEs to describe everything. So they go, it's in Fortran, but the heck, let's just put a wrapper around it and call it in the next set of programs. But they never go back and say, well, what happens when you start really pushing it to the extremes? And this was news for us. It took us a while to find this mistake. And I don't think I want to tell you how we solved this problem. I think it was, you can guess, just use Euler's method in really short time steps, right? And it's as good as anything. But none of these programs, even if they've been around for a decade and have a stamp of approval by the scientific community, you have to test them in the time regimes that you're looking at. So I think that was an important aspect, at least for me, So that's why I wanted to be sure to bring this slide up, guys. To get that time step, the communication between the CME, the Gillespie algorithm, and the ordinary differential equations. Okay.